Oops, I should probably shut that down. I think I'm on the grade book too. Yep, close that down. Oop. All right. Are there any questions before we begin? Not hearing anything. Let me open up chapter 13. And then I'll share my screen. Can, excuse me. Can somebody please confirm you can hear me?
I can hear you.
Thank you. Optimize. There we go. Should be seeing my screen now. I have that we were talking about COVID-19. Is that right? I usually have a 2 here in here. I'm not finding that. Oh, where is it? There it is. So this is slide 53. All right. I have that we had talked about retroviruses. Is that correct? There's actually 3
I
of these.
remember we had just started that. Yeah.
All right. I should get rid of these. All right, let's go back. So retroviruses have a different stage where they multiply in the nucleus. I thought I even drew this where there's paint. Let me get paint out here. All right. So the virus infects a cell and gets inside the cell where the virus takes the single strand RNA and its positive single strand. Leave that out. Have I given you this graph before? Single strand, oops, positive strand RNA. And we have reverse transcriptase. Let me get an arrow here. And I'm just going to go RT for reverse transcriptase. Have I done this? And the reverse transcriptase makes the single strand DNA. I'm sure I've done this. Can anyone confirm it?
I remember watching this, but I can't speak for everybody else in the room.
All right, so I did go through this. This is what I wanted to do this. Sorry, I've got a tickle in my throat. Hopefully you didn't see that. Um, Single strand DNA. And then the reverse transcriptase. Let's see if I can just copy this. Works on the single strand DNA to make double strand DNA. And so this happens in two steps. Your book doesn't talk about that it actually takes two steps to make double-strand DNA from single-strand RNA. And both steps are done by the reverse transcriptase, an enzyme which is in the retrovirus genome, meaning the virus carries it. There's also a retrovirus, a reverse transcriptase protein in the virus. One protein which can take the single-strand RNA, positive strand RNA, to make single strand DNA. And then this double strand DNA integrates into the host cell chromosome. And that remains a permanent part of the cell. For as long as this cell is alive, it'll have retroviral DNA integrated into one of the chromosomes. And where this integrates into the chromosome is almost at random. It's not quite at random, but it can integrate in different chromosomes in different places on different chromosomes. And that really mucks up the DNA, actually, because in each cell it could integrate in a different location, the DNA. And so I don't know, we'll put X chromosome here. And see if I can change the color here. Oh, there we go. Maybe blue would be better. And this can integrate right here, for example, the retroviral DNA. Now, whenever the cell replicates its DNA, it will replicate the retroviral DNA. But a worse trouble to the cell is this retroviral DNA can be transcribed. And when that happens, it's going to make, oops, wrong color. Well, good enough. It will make retroviral RNA, and this will serve as messenger RNA, which will then go into the, I'll go viral messenger RNA, which will then go into the cytoplasm if it's a nuclear, which is an animal cell, so it has to be in the nucleus. And it'll make the viral proteins, especially the capsid proteins. Let's just make it this way. Which will then get together with the replicated RNA, and that's one of the RNA. This is acting like messenger RNA, but it's actually positive single-strand viral RNA. And they can get together to make a new virion. Yeah, let's see if I can move down a little. And this new virion will then bud out of one of the membranes of the cell, depending on the species of retrovirus. It could bud out of the cell membrane, probably most do. It could bud out of the nuclear membrane, or it could bud out of the membrane of the endoplasmic reticulum. Any question about any of that? In which case, we'll have a new virus, which will be then can infect another animal cell. And this is sort of what I showed you here. The single-strand RNA will in two steps become double-strand DNA, integrate into the host chromosome, It can be transcribed, making positive single-strand RNA, which can be read by the ribosome to make the capsid proteins, and it can get together with the capsid proteins to make the new progeny virions. And this is showing you the big picture, which I've already gone through, so I won't go through it again. But the retroviral DNA, double-stranded, can integrate into the host chromosome. This can happen in different chromosomes in different location on the same chromosome. And when it's transcribed, it'll make positive strand, single strand RNA, which will serve as messenger RNA to make the capsid proteins. And it can also serve as the genomic RNA, getting together with the capsid proteins, which will then make a new virion, which will bud out of the cell. And that's the multiplication of a retrovirus. The one exception to the RNA viruses, which the rule of thumb is they multiply in the cytoplasm. Retroviruses are an RNA virus, but they multiply in the nucleus. And it's because they have a DNA intermediate. Any question about any of that? All right, let's move on to family coronaviruses, coronaviridae. They are a single positive strand RNA virus. They are enveloped. They were well known to humans long before COVID-19 came out because coronaviruses are the second most common cause of the human cold. Coronaviruses, besides causing human colds and COVID-19, also cause SARS and MERS. And these all three are severe acute respiratory syndrome. That's actually what SARS stands for, severe acute respiratory syndrome. MERS stands for Middle East Respiratory Syndrome. And then COVID-19 actually stands for coronavirus ID'd in 2019. Why it's called COVID-19. MERS has the highest mortality rate. Around 36% of people who get MERS will die. But MERS does not spread from person to person. Most people who come down with MERS are people who are in close association with camels. Apparently, MERS grows in camels, although it's not lethal to them, and then they can spread from camels to people. If you don't know, I am not someone who frequently spends time with camels, so I'll probably never get MERS. SARS was a coronavirus that first started in 2003 in China, and it actually did spread to the world. However, all of the SARS variants were symptomatic, so there were no asymptomatic patients that we know of. And so the medical profession were able to quarantine individuals who came down with SARS, and then they actually made it extinct. However, SARS reemerged in China once again. I don't remember the year, but it was sometime after 2003 and before 2019. And the danger of SARS is about 10% of the patients died. COVID-19 can kill patients, but when it first came out, it only killed between 0.2% to 3% of patients. So it's much less lethal than even SARS, and much 10 times less lethal than MERS, maybe 100 times less lethal than MERS. A danger with COVID-19 is that there are a large number of asymptomatic cases. Young adults and children, about 50% of the people who come down with COVID-19 will be, or at least were, asymptomatic. That actually may be higher now because COVID-19 has changed, but and so people will be having a COVID-19 infection and have no signs or symptoms of an infection, and they feel fine. They don't know they're sick. And then they can take COVID-19 to their grandparents, which would be a serious disease, or could be a serious disease. I should word it that way. And with COVID-19, uh, the elderly are at much greater risk of getting developing disease or serious disease than the youthful people. It is true that not infants, but newborns are also at risk of COVID-19, but it's not as bad as the elderly. And we'll talk a little bit about that. A danger of all of these is that when one patient became infected, except for MERS, they tended to spread it to other people. With COVID-19, when one person becomes infected, they're likely to infect 1 to 2.5 other people. And that's why COVID-19 has continued to spread in the human population ever since 2019. Any question about any of that? All right. COVID-19 actually does have a scientific name. It's called Severe Acute Respiratory Syndrome Coronavirus 2, or SARS-CoV 2. However, that name is so close to SARS that even scientists tend to call it COVID-19 rather than SARS-CoV 2. COVID-19 is closely related to bat coronavirus and likely originated from two bat coronaviruses that recombined and a pangolin coronavirus. The pangolin coronavirus only encoded about 10% of the original COVID-19, but it was an important part. It encoded for the spikes, and it was because of this pangolin spikes that COVID-19 was so infectious in humans, and also why it could spread to humans. They know this from sequencing bat coronaviruses, pangolin coronaviruses, and the original COVID-19. An intermediate animal likely had the COVID-19 and then spread it to humans. It might have been a pangolin. If you don't know what a pangolin is, it's a spiny anteater. Although the exact animal that was the intermediate is not known, but we think it might have been a pangolin. Transmission of COVID-19 is largely from droplets from coughs and sneezes, talking, singing, within a range of about 6 feet. It may spread from indirect contact via contaminated surface, like somebody coughs on a doorknob or coughs on the counter. You can pick it up, but it's not likely to spread that way. It's most likely to be breathed in, and most likely if you're within 6 feet of someone who gives droplets containing COVID-19. And usually if you pick it up from like a counter or some object, you usually pick it up on your fingers and then you self-inoculate a mucous membrane in your face. I did mention the COVID-19, oops, too far. Let's see if I can blow this up. COVID-19 is much more serious in an elderly adult. 75% or older people who come down with COVID-19, about 17% of them will die. This is obviously people who have not been vaccinated, and maybe it might not be people who have had COVID-19 before. Although I'm not positive of that because COVID-19 doesn't give strong or long-lasting immunity when you get the infection. And then as you reduce the age, even people 74, the fatality rate drops to 2.3%. And let me tell you, this 2.3% is mostly from those close to 74, not from people closer to 50. And the mortality rate of age 18 to 49 is 0.15%. Did I say that right? 0.15%. And then children, it's even lower, about 10 times lower. And that should really probably be age 1 to 17. It's 0.015. Now, it is true that below age 1, especially newborns, this rate increases, but it's only about 4.3% in infants, meaning children less than 1 year. And this is almost all among newborns, by the way. And then we look at children there, it's about 0.5 per 100,000 for both ages 1 to 9 and 10 to 14. And then it starts climbing. So children actually have the age 1 to 14 have the lowest risk. And then adolescents have a slightly higher risk than a child. And this risk will increase the older you get. But it doesn't really increase until I think it's above age 65. And then they say you're increased risk. Any question about any of that? So this one is U-shaped, but it's definitely much more on the elderly side than on the young side. And then it's really only children less than 1 year where COVID-19 is a risk. Let me shrink that down. All right, the symptoms of COVID-19 are mostly flu-like, though the shortness of breath and deaths are more common. The mortality increases with age. The ill health of the patient. Mortality is higher in men than in women, and it's higher in blacks and Hispanics than it is in whites and Asians. And this has been confirmed in the blacks, at least, and the Hispanics in both the United States, Canada, and Mexico. We find higher mortality rate in blacks and Hispanics than in whites. I'm not sure if Canada and Mexico have looked at Asians, but definitely blacks and Hispanics have it higher than whites in all three countries. And the reason for that is not well understood. The incubation period, you have your first symptoms 2 to 14 days past your first exposure and the virus gets infected inside you. Now, remember, many people, especially young people, are asymptomatic. And did I mention that that's about 50% of young people, children, teenagers, and young adults? You're asymptomatic. You have no idea you're infected with COVID-19 and you have an active COVID-19 infection, and then can spread it to someone who it could be a serious infection. The estimated death rate, I think this is the new one because that's lower than I had before. Yeah, the estimated death rate has dropped as COVID-19 has changed. Let's go on. So the estimated death rate is around 1.1% to well, that's around 1.1%. Reduced from 1.8% in the USA. Over time, COVID-19 became less lethal. That especially happened with the virus Omicron, if you remember that variant. One trouble with COVID-19 is that new variants come out. And there's actually two new variants which are increasing in frequency at the moment. And I only know one of them, NB1.8.1, which has informally called Nimbus. If you come down with Nimbus, your throat and you have symptoms, your throat will be so sore it'll feel like swallowing razor blades. So you do not want to get infected with the coronavirus strain Nimbus. Let me come back to the estimated death rate. The Omicron variant was actually a recombination of COVID-19 with the coronavirus causing the human cold. And this new COVID-19 that we called Omicron was less lethal. It was a little more likely to spread. And fortunately, since the variant Omicron came out, all the variants of COVID-19 have been descendants of Omicron. So all of the variants that have come out have been less lethal than the initial variant, but they're still lethal, especially to the elderly. One thing about Nimbus is it is not more lethal. It just makes you feel really awful when you have a sore throat. If you're sick, it's best to wear a mask, best to stay home and cough into your elbow or tissue. Any question about COVID-19? All right. Let's talk briefly about viruses and cancer. Some human cancers, around 10 to 15%, are likely caused by a virus. Now, it might be the virus gets the cancer started, doesn't totally explain the cancer, but about 10 to 15% of human cancers, do appear to be caused by viruses. Like human papillomavirus, it's well known that it causes cervical cancer and skin cancer. And then hepatitis B virus and hepatitis C virus are well known to cause liver cancers. Any question about any of that? Not all cancers are caused by viruses, like breast cancer and glioblastoma have no correlation to any known virus, and they do not appear, these two cancers do not appear to be started at all by a virus. Any question about any of that? Cancer is simply uncontrolled growth. The cells acquire new property and essentially they divide uncontrollably. Viruses appear to cause cancer in two ways. First, you can have a formation of a provirus. When the provirus integrates into the genome of your cell, it permanently changes the genetic material of that cell. And the DNA virus can actually go inside of gene and mutate it or turn the gene off. And that would be important if that was a tumor suppressor gene, the virus turned off. Tumor suppressor gene is a gene that suppresses the formation of cancer. Or the virus could integrate into the DNA near a potential oncogene and then turn on that oncogene, which can then lead to turning on a gene, which could then lead to cancer formation. The point is this DNA, viral DNA, integrating into our genome mucks up our genome, turning on genes that should be off, turning off genes that should be on, and perhaps even causing a direct mutation. Like if the virus integrates right inside a gene, that gene is going to be mutated. So formation of a provirus can lead to cancer. The second way a virus can cause cancer happens with an oncogenic virus. An oncogenic virus is a virus that contains an oncogene. An oncogene is a cellular gene involved in cell growth, but it's a mutated form of the normal cellular gene. And this mutated normal cellular gene causes the cell to grow. And that starts the cancer process. It may not be the only step in cancer, but if these cells start growing and then the cells obtain other mutations, those cells can become a cancer. And so the oncogene hits the cell, causes the cell to grow, and that starts the cancer. Any question about any of that? Now remember, an oncogene is carried only on oncogenic viruses. There's only a few oncogenic viruses, and if you get infected with this virus, you are likely to come down with cancer. Any question about any of that? All right. I mentioned that the two most common life cycles of an animal virus are the budding life cycle and the lytic life cycle. However, there are two other life cycles. There's a latent life cycle. In the latent life cycle, the virus remains in an asymptomatic and is asymptomatic in a host for long periods of time. While the virus is latent, it does not cause disease, the virus does not replicate, and does not damage the host. It just sits and then waits, and then when the virus leaves latency, it tends to enter the budding life cycle, and the virus will replicate in the budding life cycle, can damage the host, and then cause disease. Two classic cases of a latent virus are herpes simplex virus 1, causing cold sores when the virus is actively replicating, meaning when the virus is in the budding life cycle. And then the cold sore goes away, the disease goes away, the virus re-enters latency, and then just sits out, waiting for a time when the virus leaves latency, and then the virus re-enters the budding life cycle, replicates and causes damage and then disease. And that's what happens with the next outbreak of the cold sores. Another example of latent virus is the child comes down with chickenpox. The virus is in the budding life cycle, replicating, causing damage, causing disease, and then the virus becomes latent. And this virus can go latent for decades. Usually the virus doesn't leave latency until the child is now an elderly adult, and then the virus leaves latency, enters the budding life cycle, and then causes the disease shingles. The first time you come down with this virus, you'll have signs and symptoms of chickenpox, and then all the other times you have this virus replicating and causing damage and disease, it will be the shingles. Any question about any of that? There's usually a trigger for the disease leaving latency and then entering the budding life cycle, causing disease. The trigger for shingles is a stress, and it's usually just old age when the patient's immune system weakens, and then the chickenpox virus activates what causes shingles, not chickenpox. The stress for the virus herpes simplex type 1 causing the cold sores is, what do you think? What do you think the stress is? It's kind of obvious. The stress is simply coming down with a cold. It causes the herpes simplex virus to leave latency and then cause disease, which causes the cold sores. And that's why we call it a cold sore. Any question about any of that? All right. There are also another life cycle of viruses, and this is the persistent viral infection. In the persistent viral infection, the virus replicates at low numbers, usually for long periods of time, but that's not terribly important for now. And it's a naked virus, but it leaves the cell in a vesicle, meaning the virus is encapsulated in a vesicle inside the cell, and then the vesicle fuses with the cell membrane, releasing the virus outside the cell. And that's a persistent viral infection. A persistent viral infection tends to last over a long period of time. The detectable levels of the virus tend to build up, meaning increase over a long period of time. The virus may never be cleared by the host immune system. The virus can be fatal, such as in the old days when HIV caused the disease AIDS. They said that was a persistent viral infection. Another example is the measles virus causing subacute sclerosing panencephalitis. This can be fatal, and it tends to get worse over time, where initially the virus is very mild. The symptoms, signs, and symptoms are mild, very mild, but it tends to build up with time. And then with this one, it can be fatal. But persistent viral infections do not have to be fatal. They can be tolerated, like cytomegalovirus, some other herpes virus. I'll talk about chickenpox in just a minute. When a patient gets it, if they are always shedding the virus and the virus is always replicating, it's because the virus has become persistent and the patient has tolerated it. With cytomegalovirus, it usually does build up over time, but then it plateaus out, and then you're just always shedding cytomegalovirus. My mother actually had varicella zoster for the last 3 years of her life, where she came out with shingles, and the shingles never went away. So she was one of the unlucky few who had a persistent viral infection, and she always had the shingles for the rest of her life. There's less than 1 in 1,000 people who are that way, by the way. Most people get the shingles, and then after 3 or 6 months, they go away, and they're no longer shedding viruses. And the virus doesn't necessarily mean it's gone, it's just re-entered latency. Any question about any of that? All right. So when you get a cold or the flu, you get an acute viral infection where when you first become infected, the number of infective virions actually drops to zero because the virus has infected your cell, it hasn't replicated, so the number of infective units is now zero. And then the virus quickly replicates, making new virions which are released, and that's an acute infection. The best example is the case of the human cold, and then the virus quickly goes away. The host immune system repels the virus with the colds. I think also the virus itself just goes away. Because that's not long enough for the immune system to, too far. My goodness, I really went down to the bottom. Oh, there we go. So an acute viral infection where you quickly get virus, you shed a large number, and then the virus quickly goes away. No signs or symptoms because the virus is gone. A persistent viral infection, on the other hand, builds up over time, gets worse. It may bloom, and if we have a broken chart here, it means the virus killed the patient. Like this would be what happened when the patient had HIV, and then the patient gets AIDS, and the patient dies. Those are the old days before modern medication kept the AIDS patients and HIV patients alive. A latent viral infection, you'd have first an acute viral infection. The child gets chickenpox. They're shedding a large number of viruses, and then the virus goes latent. It's still around, but it's not replicating, it's not causing disease, just hanging out. And then usually when the child is now an elderly adult, you'll have the latent viral infection where the virus will come out again, shed, usually at lower numbers than the acute viral infection, causing latent disease, which in this case will be the shingles. In most cases, the shingles, the virus will go away and re-enter latency. Any question about any of that? All right, let's talk about prions. Prions is a term for proteinaceous infectious particle, where the disease is caused by an infectious protein. There is no genome in a prion. Prions can be inherited and they can be transmissible by ingestion, transplant, surgical instruments, and it's thought that they may be spread, at least among animals, animal to animal, although it's possible the animal who's got the prion sheds it, and then the other animal eats something that the shedded virus is on, like cattle or sheep. Actually, sheep's a good example. Get prion, and then they shed it, and then it hits the grass, and then another sheep eats the prion by eating the grass. Prions cause spongiform encephalopathy. Encephalopathy is a vacuole in the brain. Humans and other animals can get spongiform encephalopathy. In animals, we call it bovine spongiform encephalopathy, and a more common name is mad cow disease. Sheep scrapies, cervid chronic wasting disease, and then there's a new one, feline spongiform encephalopathy or mad kitty disease. Scrapies is the one that humans have the most knowledge of. Scrapies has been a disease that the ranchers in Europe have been dealing with for centuries. And it tends to hit sheep. Very seldom does it spread from sheep to anything else. Like I said, the ranchers in Europe have been dealing with scrapies for centuries. In humans, we don't call it bovine spongiform encephalopathy. We call it Creutzfeldt-Jakob disease, Gerstmann-Straussler-Schnickler syndrome, and I could be butchering that name, I'm not German, fatal familial insomnia, and Kuru. Any question about any of these? All of these are diseases caused by prions. Kuru is the one I know the best, and perhaps humans know it the best too. Kuru is a disease that was seen in the Fore tribe in Papua New Guinea. There's New Guinea. Let me see if I can blow this slide up. And the red is showing you where the Fore tribe was, that little region there. The patient who came down with Kuru became unable to walk. They lost their ability to swallow or chew, leading to drastic weight loss and eventually leading to death. How was Kuru transmitted? The Fore tribe had an unusual funeral ritual where people cooked their dead relatives and then ate their dead relatives. Someone in the Fore tribe came down with Kuru, and then they started eating, especially the brain and the nervous tissue of that individual, and then the Kuru started spreading in the Fore tribe. Any question about any of that? Here we're looking at spongiform encephalopathy in the brain. There's a normal histology of the brain. Kuru, scrapies, and then classic Creutzfeldt-Jakob disease. You can see holes in the brain, making the brain look like a sponge, where the term spongiform comes from. Encephalopathy deals with the brain. How are prions replicated and then cause disease? The mechanism of a prion is that there's a normal protein called PRP superscript C, which is required by the cell, especially a nerve cell. It needs to be on the cell surface, so it's a cell surface protein. And this protein can unfold and take on a different folding pattern, which is the scrapies or the prion form of the disease. The prion protein can then come up to the normal PRP superscript C protein, causing this protein to take on the scrapies folding pattern. And then the cell says, oop, I need more PRP superscript C protein because I'm a nerve cell and I need this protein for my cell surface. So it makes more PRP superscript C protein. However, the cell has two forms of the prion protein in it now. And if either of these come up to the normal protein, it causes this protein to unfold and take on the prion form of the protein. The prion form of the protein accumulates in the brain cells and then starts forming plaques, meaning the proteins start sticking together and then start forming plaques. And it's believed that the plaques form the vacuoles seen in the brain. Anyways, it leads eventually to cell death, and that will eventually lead to prion disease. Especially you lose too many nerve cells, you're going to get prion disease. Any question about any of that? All right. You might remember that in Britain, they had a epidemic of cows coming down with bovine spongiform encephalopathy or mad cow disease. And it's because the British wanted to use up the cow brains and nobody wanted to buy it and eat it. So they took the cow brains, ground them up, added the cow brains to grain, and then fed the grain to cattle. One of the cows came down with prion disease. They ground up the brain, fed it to cattle. All the cattle that ate that grain, or at least most of them, came down with prion disease, and that quickly ballooned into an epidemic of mad cow disease in England, or the United Kingdom, I should say. Any question about any of that? All right, let's move on. Plant viruses and viroids we need to talk a little bit about. Plant viruses resemble animal viruses in many ways. They cause many diseases of economically important crops, such as beans, corn, sugarcane, and potatoes. Because of the tough cell wall that plants have, the plant viruses tend to enter through wounds or via a parasite, the parasite being a nematode, a fungus, or an insect. Infected plants can then, the virus can then infect the plant cell and infect the plant. The infected plants can then spread the virus in the pollen or in their seeds, and then the plant virus can continue. Any question about any of that? We won't talk further about plant viruses because plant viruses are viruses that infect only plant cells. They do not infect animal cells, so they do not cause disease in animal cells. All right, let's move on to viroids. A viroid is a naked RNA molecule having no protein coat. You can think of a viroid as an RNA virus lacking the protein coat. So a viroid, all it is naked RNA, usually a small molecule. And this can infect the plant cell, then replicate in the plant cell, and then shed by the plant cell, causing disease in the plant. Viroids are only a plant pathogen. There are no viroids known to infect animal cells. There's no viroids known in animal cells. So for our purposes, they only infect plant cells. And here's an infection. This is a normal potato. These are potatoes infected with a viroid called spindle tuber viroid. You can see the potatoes are skinnier. Any question about any of that? Any questions about chapter 13? Viruses, prions, and viroids. All right, if not, let's move on to the next chapter. Let me go ahead and make sure chapter 14, 13 are on. Chapter 14 is already on. Chapter 13. Our last chapter is chapter 7. Chapter 7, the control of microbial growth. Always give this as the last lesson because students already have a pretty good understanding of this chapter. Controlling microbial growth will be things like sterilization, disinfection, antisepsis, degerming, sanitization, autoclaving, pasteurization, biocide, germicide. There are some terms you should know for this chapter. And then 2, be able to discuss the factors that influence microbial death rates. 3, be able to discuss the 3 main ways that cells in which antimicrobial agents inhibit or kill cells. 4, understand the various types of disinfectants and how they work. And 5, understand the relative resistance of the major microbial groups to antimicrobial agents. Any question about what we're going to do in this lesson? All right. The rate of microbial death rate is fairly constant if you graph it on a logarithmic scale. Let me blow this slide up. Meaning at time 0, when you expose the cells or microbes to an agent, and there's a million of them, at 1 minute, there will be only 100,000 left, meaning 10% survived, 90% died. 2 minutes, there will be only 10% of these 100,000 left, or 10,000, meaning another 90% died. And then that continues at 3 minutes, 4 minutes, 5 minutes, 6 minutes, where at every time you get 90% dying, or 10% living each time. And if you graph that, you'll see that the number of microbes on the log scale versus the time on the arithmetic scale will be a straight line, meaning y equals mx plus b, a real easy formula for explaining this. If we were to graph the number of cells, meaning not the log number, but the total number of cells, the arithmetic number, versus the time here, you'd get not a nice equation. You get an equation like this, something like y equals mx plus b plus, let's say, c plus nx b, no, nx, yeah, nx, or n squared x, or is it, no, it's n x squared, sorry, n x squared, meaning this is a much more complicated equation. So the bacterial population dies at a constant logarithmic rate when it's heated or treated to an antimicrobial chemical. Any question about any of that? The rate of microbial death can be affected by, number one, the number of microbes. The more microbes you have present at the beginning of treatment, then the longer you'll need to kill off all the microbes, because there's more microbes, so the longer you'll need. The environmental influence can affect the microbial death rate. The presence of organic matter can slow the microbial death rate. Now, in a hospital specimen, you're most likely going to have microbes around organic matter. And that means when you're treating the microbes with an agent, you have to expose it much longer than you would without the organic matter around. Or you must use much more antiseptic material. For example, bleach is very good at killing cells, but bleach is easily inactivated by organic matter. And so if you want to kill off the microbes and there's organic matter around, let's say somebody had an accident and had diarrhea and it went on the floor and you want to kill that up so nobody else picks up the bacteria, you have to use really a lot of bleach or expose the agent to a much longer period of time because of all the organic matter that's around. The temperature of the treatment can also affect the microbial death rate. Generally, the hotter the temperature, the better the treatment, meaning the less time you need to apply the treatment to get effective killing. And then lastly, the presence of biofilms can influence the effectiveness of the treatment. If a bacteria has a biofilm, you're going to have a much more difficult time killing off those microbes than you would if the microbe did not have a biofilm. Is that clear to everyone there? Anyone confused about that? All right, I've always mentioned that time exposure can affect the microbial death rate. You generally need more time of exposure to kill off more cells. Also, you need a longer exposure time if you have something in the microbe that allows the microbe to tolerate or survive in your treatment. For example, endospores require a much longer treatment time than bacteria lacking the endospore. And obviously, we're talking about a similar bacteria, or maybe even the same strain, a different species of bacteria, but a different strain. If I said that right. And then there are microbial characteristics which can also influence the microbial death rate. An endospore is actually a microbial characteristic. Cells that have endospores are much less likely to be killed by an endospore. Any question about any of that? All right, this slide is showing you some of the terms. Sterilization refers to the removal or destruction of all forms of microbial life, including endospores. How we sterilize things normally in microbiology is you put it in the autoclave. It's sort of like a giant pressure cooker. You have commercial sterilization. That's sufficient heat to kill endospores, like in Clostridium botulinum in canned food. Disinfection is the destruction of vegetative pathogens, usually by a disinfectant. It may not kill endospores, depending on what the disinfectant is. Most disinfections do not kill endospores. Antisepsis is destruction of vegetative pathogens on living tissue. So the only difference between a disinfectant and an antiseptic really is what you're applying the agent to. If it's to skin or living tissue, we call it an antiseptic, but if it's an object, we call it disinfection. Any question about any of that? So bleach, when you're applying it to, I don't know, the gauze you're going to put on a patient who has necrotizing fasciitis, that used to be a treatment. I don't know if it's still used, but it used to be a very good treatment for removing necrotizing fasciitis. And anyways, my point is bleach would be used as antiseptic there. But when you use bleach to clean the toilet, you're using the bleach as a disinfectant. Degerming refers to the removal of microbes from a limited area, such as the skin, before you inject with a needle. We degerm the area, usually with alcohol and swab. It is not an antiseptic because alcohol is so weak that a lot of the cells will be left behind on the counter. Sanitization refers to the treatment to lower microbial counts on eating and drinking utensils, usually in the food industry. And this is done monitored by the public health. All right, let's talk about biocide and germicides. A biocide is a substance that kills microbes. It does not usually kill endospores. It usually kills vegetative cells. A bactericide is an agent that kills bacteria. A fungicide, an agent that kills fungi. And then you have virucides, and these are agents that inactivate a virus. They may kill the virus, but they don't have to. They can just inactivate the virus. A biocide, once again, is something that kills. A bacteriostatic agent is an agent that only inhibits the growth of the microbes. Like a bacteriostatic antibiotic, inhibiting only the growth of the microbe, it does not kill the microbe. Two examples are the antibiotics chloramphenicol and tetracycline. They tend to act as well, they act as a biocide, but they're not, they're actually a bacteriostatic agent. The trouble with a bacteriostatic agent is once you remove the agent or stop applying the agent, the growth might resume. Any question about any of that? Sepsis refers to microbial contamination. Asepsis is the absence of contamination by unwanted organisms. You use aseptic technique as an importance in surgery to minimize contamination from the environment, as well as infection from the surgery room and the patient themselves. And different hospitals usually will post this information. It's especially true of hospitals that only allow visitors who have been vaccinated against a certain disease or something like that. All right, let's talk about the actions of microbial agents. Antimicrobial agents kill cells by one of three means. They can alter the membrane permeability, such as damaging the plasma membrane. This will allow the lipids and proteins and carbohydrates to leak out of the cytoplasm. If this happens, the cell could lose its DNA. The cell is obviously dead because it's losing its ingredients, but anyways, that's the first way an antimicrobial agent can kill or inhibit a cell, alter the membrane permeability. The second is damage intracellular proteins. If you denature the proteins, especially enzymes, by targeting the disulfide bridges, you can damage the membrane, forcing it to not grow and then eventually die. And then the third way that an antimicrobial agent can kill microbes is to damage the nucleic acid. If you damage the DNA's nucleic acid, oftentimes the virus of the cell will stop replicating. Any question about any of that? All right. We have physical methods for controlling microbes, including heat, filtration, low temperature, high pressure, desiccation, osmotic pressure, and radiation. I went through that very quickly, but we will discuss, well, most of them. See if I can blow that up a little. All right, heat is one method which can be used to control microbes. Heat, all methods of killing, kills the microorganisms by denaturing proteins and enzymes. Though if you flame or incinerate, you can actually turn the cells into char, and obviously that will kill the cells as well. We have two different types of heat. There's wet heat and dry heat. Wet heat, an example, is boiling, like you're cooking your vegetables, you get the water to boil. But you can also use heat in an autoclave or heat in steam. And this will, if using steam, will touch wherever you spray with the steam, and that will kill the microbes. Autoclaving is a little different, and it too kills what you give it, but you have to specifically mark what it is on your petri dish and then turn it in. And then the lab assistant will grab it the next time he turns on the dishwasher. And then the dishwasher frequently goes away, in which case you go and you look in between the trees and you can't find, I guess, a shrub, I guess you can't find anyone to run a test. Anyways, those are the two types of wet heat, boiling, autoclaving, and steaming. Very effective. You can also do dry heat. This includes baking, heating something in a Bunsen burner, and that would be called flaming. And then incineration, where you're taking the red hot loop and putting it into the Bunsen burner. And if the loop comes out red, that's because of all of the molecules. What was I talking about? Oh, we're talking about dry heat, flaming incineration. Flaming incineration turns the molecules into char, and that can kill the cells as well. It's simply baking. Baking is just sticking your ingredients into the oven to bake it. Any questions about heat? All right. Moist heat denatures, coagulates proteins and enzymes. Boiling a liquid 100 degrees Celsius or flowing steam tends to kill vegetative bacterial pathogens, most viruses, fungi, fungal spores, within about 10 minutes, often much less. Like E. coli, you don't need to have it in 1 minute to 100 degrees Celsius. Actually, E. coli will die something like around 65 degrees Celsius in about 1 minute. So some cells die much faster than others. Other cells, like endospores, and some viruses take longer to destroy. Endospores can literally last for, well, nearly ever in most endospore antibiotics. So, how do we get reliable sterilization? We use an autoclave, a giant pressure cooker that you put your media or things you want autoclaved in the pressure cooker. Turn on the autoclave. It puts pressure down on the liquid water, causing the water to flow out. I'm thinking my mother just did something. I don't think it would work, but we'll look at that later. Anyways, steam under pressure, like a pressure cooker, is very effective at killing microbes. Increased pressure raises the boiling point of water much higher than temperatures that can be achieved normally. Like the highest you can get water is 100 degrees Celsius, at least at standard atmosphere pressure. But in the autoclave, steam comes out. The temperature is usually 121 degrees Celsius in the autoclave, and that means that you can kill all microbes within about 15 minutes simply by putting the steam in the autoclave. On the other hand, if you were trying to kill, what do I have there? Something else, like Mycobacterium tuberculosis, it has mycolic acid in the cell wall. And so the mycolic acid can withstand treatment much longer, although an autoclave would kill it because it's high enough pressure, high enough temperature that the endospore would be killed by an autoclave. So an autoclave can kill endospores, and there are a few other treatments which are effective against endospores. I used to know the antibiotic which could kill an endospore, and I can't think of one other than Streptococcus pneumoniae. This endospore grows anaerobically, or at least partly anaerobically. That's probably why the friend of mine gave me their bottle of disinfectant It's because they knew the disinfectant would not kill the endospore, so they didn't really want the disinfectant. Any question about any of that? All right. We already talked about moist heat denatures coagulates proteins. Moist heat is better at killing off organisms than dry heat. And you actually know this, and that is you cannot stick your hand through boiling steam because the steam would immediately burn your skin. However, you can stick your hands into a hot oven, and we do this all the time. It's not that the hot oven will not kill the microbes, it would kill your cells too. It's just you need long enough time to kill the cells. And when I was working on the train, the pieces were probably protected for from sunlight and UV light for at least well many months. And then my roommate got it out and put it together and found that the microbes were killed by the moist heat. It's just that moist heat tends to transmit the energy or the heat better than dry heat. And that's why you can put your hands into an oven and pull out whatever you're baking. You cannot put your hands through steam. I've already talked about that. All right, let's talk briefly about pasteurization, then we'll quit. Pasteurization reduces the number of spoilage organisms. It does kill all pathogens, but it does not kill all microbes. And that's why pasteurized milk has a shelf life, because the other microbes that are not killed by the pasteurization are alive and they can grow, creating a toxin. And that's why refrigerated milk should be kept in the refrigerator and should be kept only for about a week. After that time, it's probably best to throw it away because microbes could grow in it slowly, but you have a week to grow, and enough could be grown that you now have a lump on your trying to say here, you have a lump on your dish, and the pasteurization could kill the microbes, but not all the microbes. It does kill all the pathogens, and that's why the milk can go bad. That's what I'm trying to say. And that's why the pasteurized milk has a shelf life. There are some organisms which can survive the pasteurization process, meaning they are thermoduric, heat resistance, and that can eventually spoil the milk. A little longer, the milk can survive longer than unpasteurized milk, but the pasteurized milk does have a shelf life. Pasteurization happens in two ways. You can expose milk to 63 degrees Celsius for 30 minutes, or you can expose milk to 72 degrees Celsius for 15 seconds. Most milk is pasteurized by the 72 degrees Celsius for 15 seconds. Besides pasteurized milk, we also have ultra high temperature milk. This milk is not pasteurized. What you do is you subject the milk to 140 degrees Celsius for around 1 second. That will sterilize the milk, and then the milk does not require refrigeration, assuming it's closed and sterile. And you can keep that milk on the shelf for a long period which you cannot do for pasteurized milk. Any question about any of that? All right, if there's no questions, I will end the lesson here, and I will see you in the lab at 6:30 if you have questions. If you don't have any questions, you do not need to show up at the lab. All right, any questions? All right, I'll say goodbye.